Good morning. Thank you everyone for attending this KOL call, which serves as a follow-up to our 7th annual Genetic Medicines and Cell Therapy Manufacturing Summit that took place last week. My name is Keay Nakae. I'm one of the senior healthcare research analysts at Chardan, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. Before we get started, let me read this compliance statement. Our participant has attested and agreed that they will not share or discuss any material, non-public, or confidential information that they are aware of. A recording of this call will be made available to clients after the event. Joining me on the call this morning as co-host is my colleague, Senior Healthcare Research Analyst, Dr. Geulah Livshits, and our special guest, Dr. Deborah Hursh, Principal, Hursh Cell Therapy Consulting. During the call, for anyone in our audience who would like to submit a question to ask of Dr. Hursh, please type it into the box and we'll try to ask it on your behalf. At this year's summit, we heard from 23 public and private companies regarding the current challenges and advances in genetic medicines and cell therapy manufacturing. Topics included the logistics of incorporating process improvements, clinical holds, comparability, scale-up, supply chain logistics, qualification assays, analytics, new technologies, make versus buy considerations, and to what extent regulatory standards are changing. In this KOL follow-up call, we will discuss a number of these key CMC issues from a regulatory perspective, leveraging Dr. Hursh's extensive experience at FDA and CBER. So with that, thank you so much for joining us today, Dr. Hursh. And before we get into questions, could you please share with the audience your background as it relates to cell and gene therapy CMC?
Okay, so I have a PhD in molecular, cellular, and developmental biology. I worked as a CMC reviewer in the division of cell and gene therapy. That was in various offices while I was there. For 22 years, I retired at the end of last year. And during that time, I spent half my time reviewing files in cell and gene therapy, and the other half of my time running a research lab that did research in cell and gene therapy.
Well, great.
That does it.
Well, great. Yeah, thank you. And thanks so much again for joining us today. So with that, let me hand the mic over to Geulah to ask the first set of questions.
Thanks, Kay. So Dr. Hursh, in your role as a consultant and also from your time at the FDA, what in your view are the CMC-related issues that tend to trip up sponsors the most? First with their initial IND filing and then also at later stages, such as they get ready to file their BLA.
Oh, okay. So the office that is now OTP, was OTAT most recently, offered a lot of pre-meeting opportunities. There were the interacts, there were the pre-INDs. The reviewers spent a great deal of time on those, trying to foresee what sponsors would run into. And then the IND would come in and the sponsor had not implemented any of those requests. So that usually caused a problem, plus it was exceptionally frustrating for the reviewers. So I would say, you know, if the FDA is giving you meetings and they give you extensive comments, you should try to implement them or at least address why you couldn't implement them. So that's one thing. Another thing for allogeneic products was donor eligibility. It was, you know, oh, did I have to do that? And that tripped people up because if your cells don't meet donor eligibility, then you need to apply for an exemption, and essentially that backs you way back up again. And it's a very uncertain thing. You may or may not get an exemption, and the timeline is exceptionally uncertain. So that was another one. People didn't seem to understand that vectors were considered critical components of manufacturing and needed to be under a quality system pretty much from the beginning, that you couldn't just make it on the bench. That was another one. And then another one is to come in with your IND and not show you can actually make your product. You should have run some engineering runs and show that you're capable of making this stuff before you come in for an IND. The other one, and this trips people up all the time, is not having the reagents you're using appropriately tested. Every reagent that goes into your product should be tested, or at least you can document it's been tested for adventitious agents. A lot of people come in and say, oh, well, I have a GMP grade product or reagent. There aren't any GMP reagents. That's just, it's not a grade. So something made under GMP may not have been appropriately tested for adventitious agents. So again, that backs you right back up again. And then for BLAs, probably the biggest problem is that people make manufacturing changes, really large manufacturing changes. And that's inevitable. I think the agency understands that's inevitable. But you just have to understand that's going to slow things down. You need to build time in for that and think about that. The other thing is that while you don't have to have validated your assays prior to, submission of your BLA, you should be able to demonstrate they're capable of being validated. And a lot of times people will come in with particularly potency assays that the agency looks at that and says they're never going to be able to validate that. And that'll be a problem too, because if you can't validate your assays, you can't get your BLA. You would really rather not go into your phase 3 with a bunch of assays. You'll never be able to take them through formal validation. So I think those would be the things that I've seen that, cause things to sort of blow up in a sponsor's face.
Mm-hmm. That's really helpful. So maybe we'll come back to the process changes later, but I'd like to talk a little bit more about the analytics and assays that you were just alluding to. And this came up as a recurring theme in our conference last week, the importance of analytics to ensure the consistent quality, efficacy, safety, reproducibility, et cetera, during the entire manufacturing process. So can you talk a little bit more about potency assays and why this has proved challenging and how that is reflected in kind of the different buckets within the cell and gene therapy space?
Sure. And I acknowledge potency assays are probably the single most challenging thing. People are always trying to figure out how to be a device because devices don't have potency assays. But the one thing is that a potency assay should be very specific for your product. It should be testing the action or actions your product is supposed to affect to get a response. And so people talk a lot about platform potency assays. I have a hard time seeing that working. I have found that sponsors, once they get a good potency assay, are really willing to share it with people. So not only is it something that's hard to develop and probably covered by IP, but it should be very product specific. So a payload specific thing for each of your gene therapies. So they need to be quantitative. They need to be suitable for a manufacturing environment, which means that, it can't be something that only the prize postdoc in your research lab can do. And so they shouldn't really be super complicated. They should be something that can be validated, is fairly robust. The other thing is that the potency assay is used for a lot of things other than releasing your product. So your stability program will use your potency assay. Your shipping and handling assays will use your potency assay. So your comparability will certainly use your potency assay. So this is something you really need to invest a great deal of time in. It's probably something you should be working on, from the moment you sort of think about, this is what I think my product does. The other thing sponsors don't often think about is that your potency assay may have a bunch of components to it. So an example that I remember was, there was a gene therapy involved, but it was in a specific type of cell. And the potency assay had to both reflect the activity of the transgene, but also what those cells were. So it was like a 2-component potency assay. It was very complicated to develop. So, and in our potency assay guidance, which admittedly is there, I don't work there anymore, the potency assay guidance, which is admittedly a bit old now, they talked about what they called the matrix approach, which was that you might have a bioassay that was very indicative, but not very quantitative. And then you would add in a bunch of physical assays that could be very quantitative. And you could kind of package that together and get something that was very potency indicating. So I think those are probably the things I saw the most.
And just to follow up on that, in terms of the type of assay that could be validated, can you elaborate a little bit on what that means?
Well, when I say validation, I mean the full-blown ICH Q2, intermediate precision, robustness, ruggedness, do it in 2 different places by different operators. I mean, showing linearity. This is a whole process. It takes a long time to do a full validation, and every assay you send in should be validated unless it's some sort of compendial thing. And so there are certain assays that you just look at them and you say, I have no idea how they'll ever validate this, because they're super tweaky kinds of assays. So it could be any kind of assay. I mean, we're not prescriptive. If you think this assay works and you think you can validate it, that's on you. But we may want to see, the FDA may want to see data before you enter into phase 3 that is even feasible.
Got it. And so in terms of the challenges for specific types of therapies, let's say CAR T, gene replacement, other types, are there any kind of unique challenges across those different buckets?
Well, I think they all have their unique challenges. I wouldn't want to speak to specifics on that. They all have their unique challenges. I think, the main thing is to start with a whole pile of assays that you're playing with, when you first start working this up, and then, slowly, decide which ones are going to work for you.
Got it. Okay, so if you want to purity, what about that? Is there guidance on testing for process and product related impurities like nucleic acid, viruses, minor cell populations, all of these things? How should sponsors be approaching that?
Well, I mean, biologics by regulation have to be safe, pure, and potent. Therefore, you can't have impurities in there. There's very little prescriptive information on things. The gene therapy guidance has some cutoffs that they recommend for, DNA and things, so cellular DNA left over from manufacturing. But this is, again, where it comes in. In your IND, you're going to have a preclinical package in which you've submitted, all this data on something which is hopefully comparable to what you're putting into the clinic. And it will establish that the impurity levels are safe, okay? And then it's up to you to make sure what you go into the clinic in has similar impurity levels. And surprisingly, this does not always happen. And it's a problem. So, and then the other thing is to sit there and look at your manufacturing and say, okay, this is an impurity. These are impurities. I must measure them. I must establish either by literature or by my own preclinical data what's a safe amount of them. I am unaware of some sort of hard and fast rule, for most of these things. It's what you can demonstrate, and keep in mind, it's going to depend on, route of administration, how much of this product are you putting in. It really depends.
Got it. And are there any other particular issues that sponsors run into on the purity assessment front?
Well, I mean, I'm not saying it's not a problem. It is a problem. I think what I found is a lot of them didn't think about, if you're making something and you have residual cytokines or residual fetal bovine serum or any of these things, it's up to you to measure them in your final product, or at least go through, for early stages, often it was allowed that you could go through extensive washes and do rough calculations that this should be gone, but that's only really good in the very beginning. You should be thinking about, okay, these are my residuals. This is what I need to assay for. And then I will either have data from my own farm tox study or from the literature that we're at a safe point.
Got it. Great. So maybe I'd like to ask a couple of questions focused on cell therapies, where you can have autologous cell therapies and donor-derived and iPSC-derived, et cetera. So for autologous cell therapies, what's the biggest concern that FDA has on that front?
Well, so autologous cell therapies are a challenge because humans are outbred. We're not mice. And so the inherent variability in human populations is enormous. And therefore, making, a product that meets sort of set lot release criteria when you're making every lot from a separate outbred human being, that's a high bar. And making cell therapies is, particularly ones derived from pluripotent stem cells, is not an exact science yet anyway. So there's inherent variability in doing the step differentiations you need to do. And then you introduce the fact that each lot is a different genetic thing. This is challenging, which is not to say that people aren't seeing some success with this. I think it's probably doable, but you just need to understand that it's going to be more challenging than an allogeneic product, though, you won't have, the fact that you need to immune suppress and things like that. So that will give you other benefits. So usually the FDA wants to see, that you have made products from several individuals, like in your pre-IND package, so that we have a sense that you have enough control over manufacturing that your biggest problem is in human variability. So that's the problem with autologous. So with allogeneic, it's still the same sort of problem. If you have to change donors, it's essentially the same problem. You have to demonstrate that your donor, your second donor is giving you a product similar to your first donor. And what I've found, particularly with pluripotent stem cell donor products, is people don't want to change cell lines. They just don't want to do it. They've got something working. And, I think the data in the literature is pretty clear that different pluripotent cell lines differentiate differently. It's just sort of a fact of life. So, so it's an issue. Is that okay?
Yeah, so I just coming back to the autologous cell therapies, I guess, what are the key QC metrics and release assays that are important to kind of to assess that variability and kind of make sure to the best of the sponsor's ability that they're making the same product from different patients?
Well, again, that's just not one size fits all. It's going to be dependent on your product. I feel like sponsors should have not only the assays they're going to call lot release assays, but they should hold in their back pockets a whole bunch of other characterization assays, so that if they need to say, these two things are sufficiently similar, they can do that. There aren't any set assays that are applied to autologous versus allogeneic, with the possible exception that you need to make sure with an autologous therapy that you're giving the right product to the right patient. Not that we wouldn't ask for identity assays for allogeneic them the FDA would ask for identity assays for an allogeneic product, but it's extremely important to have, you know, something like an STR assay for an autologous product and a whole chain of custody to make sure that the right stuff's going into the right person. I hate to say this, but slip-ups do happen, so.
And then just lastly on this, for a program that are derived from something like immune cells, what are the key FDA is concerned about with respect to things like cell identity, genome integrity, this type of issues, and anything else?
Oh, okay. So I have to say, Geulah, you're breaking up periodically. So what you want to know is about pluripotent stem cells? Okay. All right. So most of the things that are concerns with pluripotent stem cells are generic. They're not unusual. You know, we apply sort of the same ruler for most things, with the exception that, you know, pluripotent stem cells are essentially pluripotent and can divide forever. And that comes with some inherent issues, one of which is that it is pretty well understood that they do have some genome stability issues. You know, there's a lot of robust publications on that, a lot of research groups looking at that. So, so we're probably a little more concerned about that, particularly in instances where the agency thinks that your mechanism of action is that the product is going to stay there for a really long time. You know, so for things like MSCs, they're sort of like, you know, cytokine delivery vessels in general. They're not expected to hang around. But pluripotent stem cells are true regenerative medicine products. They're usually expected to integrate and do something for a long time. So, you know, any problems with their genome is going to be a concern. And I think that in general, the agency would like some assays, you know, sort of whole genome assays on the starting cell bank to show that it doesn't have any inherent mutations of concern. So for example, P53 has been seen. It's a mutation definitely of concern. And then the other issue is that pluripotent stem cell derived products are in culture for a really long time, an inordinate amount of time, longer than most cell therapies. And there can be culture in not necessarily induced, but selected mutations, something that may be in there that's being selected over the time and culture. And so I think that it's probably prudent to take a look at the end of your manufacturing and do some whole genome approaches there, to look at, you know, is there something that's coming out? You know, in some cases, you might have a problem in your cell bank, but your differentiation eliminates it. In other cases, you might have a minor problem in your cell bank, but your differentiation magnifies it. You want to know that. You don't want to find that out later. So I would say that in and of itself is my greatest concern about pluripotent stem cells.
Great. So maybe now I'll hand it over to Keay to ask some additional questions.
Yeah, thanks, Geulah. So, Dr. Hursh, let me ask about the issue of comparability. And, you know, obviously companies sometimes do need to modify their process during clinical development, either to increase process robustness, or, you know, oftentimes it's something that entered the clinic from an academic setting, and now we're looking to scale it up. So with respect to process changes, what should the sponsors be doing to minimize the extent of disruption or delay?
Oh, again, comparability is really one of the most challenging things. And, you know, it's not like the agency doesn't expect you to make changes. Of course they do. Everybody knows this is going to happen. I think what I've seen is sponsors just don't build in enough time for this. They don't really understand how complicated this is going to be, because if you are pretty late, you know, all right, in phase 1, phase 2, you know, we wouldn't even use the term comparability. We would talk about similarity, and you could probably, you know, do a fairly truncated analysis and get away with it. But, you know, if you're entering into a registry trial, we used to call them phase 3s, now we call them registry trials, and you want to make a major manufacturing change, then you'll probably be asked to go through a full-blown comparability analysis. And that is actually an analysis that requires statistics, and it will be given to FDA statisticians who are very good at their jobs to analyze. So I would suggest that you have enough samples so that you can do statistics. If you send in data that can't be analyzed statistically, they'll probably just turn it around and send it right back to you. So that would be the first thing. And again, I don't think sponsors really understand how many, you know, runs or batches or whatever they're going to need for a statistical analysis. The other thing is that you will have a suite of, you know, what are called lot release assays, assays that you release the product on. Those will not be sufficient for a comparability analysis. This is where those characterization assays I talked about come in, where, you know, you may not reveal these to the FDA. You know, they may be things you're holding off on the side, but you should be able to bring other assays to bear that augment your lot release assays to show that, you know, whatever process change you've made is not really impacting what you consider your critical quality attributes, which hopefully you've actually figured out. That's another issue. If you don't know what your critical quality attributes are, you're never going to be able to demonstrate comparability. I have seen sponsors over the years actually do small animal studies and not just in vitro things. So, you know, just to really kind of bring home that this manufacturing change does not have an impact on what we think the most important assays are. And again, you have to have put time into all those assays that they're sufficiently well developed. The other thing I've actually seen sponsors do, which always struck me as amazingly foolish, is that they will not keep adequate retained samples. You know, I had a sponsor once who actually gave all of his phase 2 material to somebody else to run another trial. And it was like, don't do that. Don't do that. Because again, later, he didn't have his own phase 2 material where he had a tiny little efficacy signal to compare to his manufacturing change. So, you know, keeping enough sample around so that you can show that pre and post changes are, you know, the same. And for an autologous product, you're going to actually have to, you know, make things from the same person. You know, so, you know, if it's an apheresis product, you have to do a split pack apheresis so that you have material from both, you know, for both runs. It's really complicated. It requires a lot of planning. It requires a lot of time. And then the agency, of course, wants to review it, which adds further time. So I think sponsors just don't get how much this can delay things. So I think that's probably my best output on that.
Well, let's maybe talk about the assays and the, you know, critical quality attributes, especially in situations where as you're going along the path of development, you may be starting out the exact mechanism of action may not be known, and hence, as you mentioned earlier, perhaps they're using a matrix approach for potency, you know, with qualitative and quantitative bioassays, and which almost by definition then becomes iterative as updated and supported by the clinical results. So how do you demonstrate comparability when your release assay specs have changed?
Oh, yeah, that's a further challenge. So to address the don't know the mechanism of action, I would hope you would at least have a hypothesis. If you don't have a hypothesis, you probably have a problem. And you may have more than one mechanism of action. So there's that. Yeah, you know, if you have super broad release specs and then, you know, you want to run comparability, that'll be a challenge. You know, you may not actually be ready to get there yet. And this is again where all these other characterization assays come in. You bring these other assays to bear to show that, all right, yes, we don't have all this nailed down, but we can show you with a huge suite of assays that this is performing the same way. You know, there are not specific assays. There is not specific limits. This is not a one-size-fits-all thing. So it's really hard to be prescriptive about this.
And I guess maybe the big question is, you talked about earlier and about how we actually demonstrate comparability. Some choose to augment in vitro with animal studies, but at what point does the changes, whether for process or CQA, require additional clinical data?
Well, you know, in the instance you just mentioned, where, you know, let's say the phase 2 has really, really broad specs, and then they want to narrow them and go into phase 3, that's a pretty high bar. You may find you're doing a bridge. You know, I've seen people required to do bridges for relatively minor changes, but it turned out that, you know, the one thing they were changing had a huge impact. So, you know, and really that decision will be sort of a collaborative decision between, you know, the CMC, the PT, and the clinical reviewer. That, you know, this, you just can't do this. You're going to have to do some sort of a bridge. Another situation that sometimes arises is, and you know, with certain cell types, and this is like ex vivo gene therapies, cell therapies, a process change will actually change sort of what the product actually is. You know, by flow cytometry, it had these markers and now it has those markers. You may find that you have a different product, you know, because if it gets that different, it's a different product. So, yeah, I think that this is just not something one can be prescriptive about.
Okay, great. Well, maybe let's go back maybe to a higher level. And based on your experience, you know, what we see sort of in the field is, you know, the science and technology continues to advance. So especially for, you know, cell therapies, where we maybe started out with autologous and, you know, pluripotent that went down a path of differentiation, but, you know, that was it, or maybe even an MSC that was unmodified, to where we now have a lot of adopted cell therapies and iPSCs that introduce a lot of edits before they go down the path of differentiation. So with that sort of being the trend and where the science is going, do you see a commercial future for the unmodified cell therapies?
Oh, that's an interesting question. So it's important to keep in mind, you seem to talk about pluripotent stem cell derived products as if they're a done deal. They are in no way a done deal. I don't think you'll find that we have actually, the FDA has not approved any products like that. And, you know, drug development is really long in general. You know, you may think CAR-Ts appeared overnight, but that's not true, and people are kidding themselves. CAR-Ts have been in development for over 20 years. So you go into the clinic with a product that you're locking down, which may be old technology, but you locked it down, and that's what you're going forward with. You may be bringing up your 2.0 behind it, but, you know, so I just want that perspective, as if all of this is changing that fast. It isn't. But I take your point about the fact that ultimately we'll get to a point where it will be routine to genetically modify any cell therapy. And, you know, when I put on my sort of futuristic hat, I say, yeah, that's probably going to be true. But we're not there yet. We're absolutely not there yet, because gene editing is, it's pretty amazing, but it's far from completely understood and developed. And the assays to really assess gene edited products, again, they're coming along at a rapid rate, but it's not like we can just say, oh, sure, this is fine. So I think that I agree that the technology that's coming online is remarkable. In the time that I was at the FDA, we went, we had sequencing, whole genome approaches, metabolomics, new optical methods, machine learning, it just went on and on and on. And there is a point at which that floats all boats. Okay, that's improving it for everybody. But if you keep implementing new technology into your product in development, you're making big changes. So, most sponsors don't really want to do that. They get someplace, they lock it down. And that's why in some regards, you'll get products that seem like, well, that was just approved. And that seems so last decade, but it's been in development for a decade, really.
So let me ask the question maybe a different way. Of the different emerging therapeutic modalities, such as gene therapy, gene editing, autologous cell therapy, iPSCs, which of these has the least burdensome regulatory pathway to commercialization due to their associated CMC issues, in your view?
Ooh. That sort of suggests that there are different regulations for that group, and I don't necessarily see that. Those are all what we call 351 biologics. They all are subject to the CFR 600 regs, as well as all the rest of them. So I wouldn't necessarily say any of them has a different regulatory hurdle. I may not be understanding your question correctly.
Is the FDA more up to speed on the critical issues for one of those therapies than others, and therefore, at least once the company has done the trials and let's say they have efficacy, they're least likely to run into a regulatory CMC issue?
Well, I wouldn't say that, but it is undeniable that the more the agency has seen a product, the more what their comfort level is better. What we ask of a first in human is usually a lot more than after we've seen the second or third or even the 10th IND. So I take your point there. And I have to say that, how many BLAs are there for CAR T cells right now? The agency is very comfortable with CAR T cells, but those are primarily the CAR T cells that are built on CD19. And eventually you'll move into other targets, which is of course beginning to happen. And that will again raise the concerns. So, pluripotent stem cells, yes, the agency's got a lot of expertise in pluripotent stem cells now, and it's relatively comfortable with them as the starting material for products, but it's early days there yet. It's really early days. Yeah, there's a huge amount of experience with MSCs, but getting MSCs to actually demonstrate efficacy and, has been a challenge. So, and that's more of a clinical issue. So does that address your question?
Yeah. Thank you. Let me ask you a question in terms of regulators' interaction with sponsors, specifically Type A and Type B meetings. We know the FDA provides minutes to its formal meetings to sponsors, and so can you tell us how much detailed feedback does the agency provide in these meetings? And is there really any reason for the sponsor and the FDA not to be on the same page regarding expectations following these interactions?
Well, okay, so a large part of my workload in the last 5 years was interacts and pre-INDs. And my opinion is that there's no reason for the sponsor not to be on the same page as the FDA, with the caveat that sometimes what the FDA actually means when they say something might not be clear. I grant that that can be confusing. There is sort of FDA speak, but you know, the agency, the reviewers, they would put in enormous amount of times in these meetings. And, you know, you're allowed to ask questions of your CMC, your farm talks, and your clinical, and you should ask good questions. You really need to ask good questions. You need to think through, what are the hurdles I perceive, and get detailed information on those hurdles. The number of times I got questions that were, does the agency think that my manufacturing is okay? You're going to get a question, you're going to get an answer of no. You haven't given us enough information to make that sweeping a statement. The FDA is very cautious. They're not going to say, Domini, your manufacturing is okay. That's just not going to happen. But if you say, okay, I have a cell line that I'm doing XYZ and I'm using these tests, do you think these tests will be adequate? Then you might get a much more granular answer. So part of this is the sponsors need to ask better questions. That was definitely something I saw. But the other thing is that, you know, you get a lot of detailed comments and you should read them. And I really think in many cases, if there's something you don't understand, you're allowed to reach back out to the agency and say, okay, I'm a little unclear what my reviewer actually meant by this. Could I have a short interaction even by email to ask some questions? I certainly always did that sort of thing. And, you know, one problem right now is that the agency, particularly the new OTP office, is exceptionally busy. It's understaffed and it's exceptionally busy. So, you know, it may be more difficult to get on people's schedules, but in general, that kind of assistance has been there. And I assume it will still continue to be there.
Okay, great. So you talked about asking questions, and a lot of our listeners are investors in early stage companies. And so, you know, in terms of your experience, both at the agency and now as a consultant, what are some of the key questions they should be asking their early stage companies, you know, in terms of the common CMC mistakes that you see them make that you're certainly now trying to persuade them not to?
Well, so if I were looking at a company, you know, it would be, okay, what is this product and how do you think it works? And what data do you have to support that it does what you think it does? How are you intending to label this product, you know, in a more regulatory sense? You know, Wilson Bryan, who was my office director for many, many years, used to say, start with the label and work backwards. And I actually think that's excellent advice. What are you going to try and sell? You know, what activity is this thing doing? And can the company say, yeah, we've shown it does this? You know, both. And then the next thing is this company prepared to make this stuff? You know, can they actually demonstrate to you that they've figured out how to make this stuff at the scale that they're going to want to sell it at? Because that's another issue is, yeah, we can make it, but we'll never be able to scale it up. So what's the business model? How are you going to do this? So those kinds of questions. You know, have you secured the places that you're going to need to manufacture this stuff? You know, for viral vectors, there's huge lag times before you can get space in a contract manufacturing facility. Have these people even considered that? And are they on someone's list to get in somebody's manufacturing facility? There are critical reagents that are really hard to come by. Cytokines, sera, there's all these proprietary media. Has the prospective company, have they secured, agreements with people to get this stuff? And will they be able to get it in adequate amounts to run their entire trial? Because you really don't want to change a critical reagent, in the middle. And it's not an FDA question, but I can't help but say that we didn't used to sit around and ask these questions among ourselves. It's like, how are they ever going to find a market for this? What is the market? And there are some very weird aspects to this, which are, again, not FDA questions, but how are you going to get this reimbursed? Is there going to be some oddball situation where this is sent to a physician's office and the physician has to pay for it before he gets reimbursed, which will be really a disincentive to the physician adopting this? Thinking about those kinds of delivery issues. So I could go on and on and on, but those are things, we look at these, they, used to be me, looked at these things and we would just scratch our heads. It's like, this is great science. This is really interesting. I have no idea how anyone's ever going to make any money doing this. And, I had to tell new reviewers that ultimately this is about people at least making some money. This is not a charity thing. So anyway, that's my viewpoint, which is very personal. So.
Great. Well, let me hand the mic back to Geulah for any final questions or questions from the audience.
I do see a question from the audience regarding whether there are standard times and conditions for stability testing. Do you have any thoughts on that?
No, there are no standard times and conditions. This is not monoclonal antibodies where there might actually be standard times and conditions. You will have come up with some idea of how stable you need your product to be. If your product is some sort of autologous cell therapy that's delivered to the clinic right after you made it, not cryopreserved, it may only be stable for 4 to 8 hours. Where if you're making an allogeneic product that you actually cryopreserve, it could be stable in liquid nitrogen for a year or more. So that's very tailor-made. It's not specific, but there is guidance on how to do stability testing. There's a lot of guidance on it. And you should be setting up a stability protocol very early on, because you're going to want to trend data throughout your entire clinical study. This is where you need these assays, because those will be part of your stability study. And it's like, oops, I forgot to develop a potency assay, but I have all this stability data. Well, have you looked at your potency assay and your stability data? Whoops, that's a problem. And the other thing is, and I have seen this happen, is you need to actually show that your assays are stability indicating. So you need to have assays that show when your product fails. All right, leave it on the bench for 4 hours and, do your assays and show they don't work anymore. But don't come in and say, well, we have no idea if our product ever fails. Well, then how do you know your assays are reflective of anything? So I guess, start early, think about a lot of assays, develop your lot release assays so that they'll be useful. And then, you start small. You may say the product's really only good at the clinical site for 4 hours, but then you keep testing. And often sponsors are able to actually get that out of there, get further. They show that the critical quality attributes are good over a longer reach. And don't forget, all the aspects like, you're shipping it, or, even if you're driving it cross town. There are a lot of things that are made in cell therapy lab A and then driven across Boston to hospital B, and that's still shipping. You need to show that what gets to the hospital is still the same stuff. It works the same way. So, okay.
Great. I think I have no further questions at this point. Kay, we'll bring that back to you then.
Okay. Well, let me ask you one final question, and this is one we often hear or hear get asked of the sponsors, and that is, how up to speed is the FDA on the latest technologies and potential implications for CMC?
You know, I think people underestimate how up to speed the FDA is, okay? I can speak mostly to CBER because that's where I worked. CBER has 100 principal investigators running research labs who are investigating assays and equipment and things that are related to this in vaccines, in blood, and in regenerative medicine. You know, I had a research laboratory. I had access to state-of-art equipment. They gave me new equipment all the time. So, and the whole point of having those laboratories, because they're very expensive, is so that there is staff on site who have direct experience with the newest assays. You know, right after Fluidigm came out, we had one. You know, we have 10X, we have Illumina, we have mass specs. They had all this stuff, and they had a lot of experts in this. It used to be that, you know, the CBER research labs were on the NIH campus, and there was a lot of cross-fertilization there. So I think people really don't appreciate that the FDA is quite up to speed, and CBER ran, as well as CDER, ran amazing seminar series, bringing in top flight people to talk about new technologies. We had a whole conglomeration of seminars the last year in 2022 on advanced manufacturing, just all the new techniques with advanced manufacturing. And this was a whole area that there was a work group that was continually looking for people. And you do need to appreciate that reviewers are scientific professionals. You know, we didn't pull these people in off the street. Your CMC reviewers in general all have PhDs or extensive experience in some specific area. All the clinical reviewers are MDs with extensive experience in clinical trial design. So these are all scientific professionals who, you know, go to meetings and keep up with the literature. So I honestly don't think you need to worry about that. You know, of course, there can always be an assay that somebody hasn't seen before. And usually the agency will reach back out to a sponsor and say, all right, this is relatively new to us. Can you give us more information? But it's not like we don't have people who, if you give us a protocol and data, aren't going to be able to understand it. And, you know, I have to say, I often was a little frustrated in meetings that people, you know, felt they had to explain things to me that I really already understood. So.
Okay, well, very good. Well, we've reached the end of our time here, but I want to thank you, Dr. Hursh, so much for joining us today, as well as our audience attendees. And again, we'll have a replay available later today or tomorrow. So with that, let me bring this to a close and thank everybody for joining us today.
Okay. Well, thank you for your time and attention. This was interesting.